Thanks again, Veronica, for the invitation. Holobionts are, I think, one of the things I enjoy thinking about the most. I often find myself sort of coming back to them in odd moments, like in the shower, for example. I don't know, maybe other people also find that they have good ideas coming in the shower, or on when I'm biking around or getting distracted, daydreaming. So it's really a pleasure to be able to think about them together in a bit more of a deliberate way, not just in my little idle moments here and there. I'd like to start with a small story about hand taste. And it's a story my colleague Kim, who's here today, actually, told me some years ago when I was doing my PhD fieldwork and getting to hang out with a lot of really interesting people and ask them lots of really annoying, stupid questions, sometimes for many hours, as was the case with Kim, who I think, indulged me for like 3 hours in a row. And one of the stories that he told me was this. And Kim, please jump in if I get it wrong. Kim used to live in Japan. He lived in a small village in Hokkaido for 4 years. And once a year, every year, all of the village obachans, the grandmothers, would come together, often with the younger generations too, at the village community center to make miso. And they'd bring all of the soybeans that they'd grown that summer, cook them together in one big kitchen, and combine them with koji and salt according to their different family recipes, and ready them to ferment for the coming year, for the coming many years, depending on what they were going for. And Kim recalled to me how these grandmothers all welcomed him, of course, how they were really excited to share all their misos with him. One of them, he recounted to me, was 15 years old. That's pretty not super common for a miso. Not all of them are so old. And apparently this one was super deep, rich, intense. And Kim told me how the flavor of just the tiniest tip of the edge of the spoon lasted on his palate for like half an hour. Amazing. I want it. And he even met some neighbors, and I think this is where it gets really interesting when we want to think about hand taste and holobionts. He even met some neighbors in this village who, using the same beans, the same recipe, the same aging, living only meters away from each other, offered him misos that still, somehow, tasted completely different from each other. And they said to him, "Your hands make your miso." And that's hand taste. Now, probably some of you in this room have heard about this concept before. Some of you have even written about it, like David, for example. I want to talk a little more about this concept. It's super interesting, especially when we start to link it to the idea of holobionts. I think that's where things start to get particularly fascinating. Now, hand taste is originally from Korean. The Korean phrase is sonmat, and originally it doesn't have to do specifically with fermentation, actually. It's sort of come to connote fermentation often nowadays, but I think it's important to remember and honor its origins, that it's actually a more general phrase. It is used often in like a culinary context to refer to the particular quality that a particular person and their life history and their experience and their skills and their preferences bring to the particular flavor of a dish. And if someone cooks particularly well, then you might say about them, they have particularly good hand taste. So it has this kind of broader metaphorical quality to it, I think is beautiful. And then, of course, when we think about it in terms of fermentation, there's a sort of extra microbiological layer, somehow also a more literal layer that comes on top, which is about how your actual hands add something to your ferments, microbes, namely. And that's what we're going to talk a little bit more about today. I guess we've already been talking about it. So this is what I'm going to try to do. And I'm going to try to do it in a completely optimal balance of speed and not too much speed. Because I want to make sure we have time for the panel, which is probably going to be I really want to get into that and talk all together about it. But I also am going to try to not talk too fast. So if I start to talk too fast, just flag me down or so. Yeah, thanks. I can get worked up, when I get excited. So I'm going to start with a bit of context. I think to sort of account for myself and how I've arrived at this, because I think there's so many different ways into this messy, fascinating, complex world of holobionts. We've already heard about 3 very different ways, so I'm going to try to briefly offer a little story about my way into it, and hopefully that can contextualize a little bit of the way that I'm trying to ask some of these questions. Then I'm going to share some examples of hand taste, particularly at this intersection of science and craft. And based on that, I'm going to get to the meat of it, what I really want to talk about, which is a framework that I've been thinking about and trying to develop for the last few years about what I'm tentatively calling holobiont hand taste. And that's this idea that I'll come to describe about how thinking hand taste in terms of holobionts can expand it, make it more complicated, maybe also more illuminating. I'll briefly touch on the role that art can play in all of this, because holobionts can be a bit confusing sometimes and kind of unintuitive, strangely, and difficult to work with. And that's where art actually can be really powerful to help us sort of encounter them through our senses and our bodies, and not just through our ideas and our minds. And then I'll try to bring everything together, of course, perfectly and successfully, with a brief reflection on method at the end. And we're going to do all that in optimum speed. All right, let's get into it. A lot of this story for me starts at this place called Nordic Food Lab. It used to be a nonprofit research institute founded by Noma and Klaus Meyer. We had the mandate to investigate the edible biodiversity and gastronomic potential of the Nordic region, and to share that information and knowledge openly with chefs, restaurants, academics, and industry, and anyone else who would be interested. A lot of that work looked like this. We made a lot of stuff. And when I came, this is some years ago now, 2012, I was also just learning and tasting so many things I'd never tasted before, and so many of the new things that were being made at that time were fermented. And what I became particularly interested in was this approach to fermentation, which I sort of since started to think of as a kind of translation, in the same way that we can translate a book from one language into another. We can also translate recipes or fermentations from one bioregion into another. And that's exactly what I saw happening here. So, for example, making misos, but instead of using soybeans and rice, which would be common in Japan, using yellow peas and pearl barley, really sort of old school, gammeldags, dansk ingredients. Now, the main goal here was flavor, to try to diversify, make new forms of flavors that could be used on menus and help to bring more flavors, more microbial biodiversity into our daily lives. But what I also started to realize when learning more about how quickly a lot of these microbes change and adapt to environments is that it was very likely that these translated fermentation experiments were maybe having much larger effects, maybe not bringing only new flavors into the world, but maybe even new forms of microbial life. And this would be happening, importantly, without these chefs and researchers even trying to do that or even necessarily realizing it was happening. That was super interesting for a variety of reasons. So that sort of became the seed of my PhD, which I then went into a cave for 5 years to do, and somehow managed to come out the other side, partly in thanks to all of the people that I had interesting conversations with, like Kim. And the short answer is, that is happening. The long answer is, it's complicated. If you're interested, we can talk more about it. But the misos became a particularly sort of illustrative point to show that that was happening. There was this sort of flavor-driven diversification happening. And now with my new group, for the last, it's not so new anymore, the last 2.5 years or so, I'm trying to bring together these different worlds, cooking, culinary R&D, natural sciences, and social sciences, to bring together cooking, science, and culture to help make the food system more diverse, more delicious, more sustainable, while also hopefully doing some novel transdisciplinary research along the way. And that's how I myself have come to holobionts. So now I'm going to shift to talk about some examples from the intersection of science and craft. this is where I'll start. One thing that I think is particularly interesting about these mesos from a holobiont perspective is that we found quite a bit of Staphylococcus species in them. Now, there are some Staphylococcus species that are also part of traditional mesos, so that's nice. Another thing I would say immediately, especially for the microbiologists in the room, is that don't worry, there are no toxins in them, there are no toxigenic genes in them, they're all safe. Just had to tick that box a little bit. One species in particular, Staphylococcus epidermidis, is usually associated with the human skin microbiome. We have to change the microphone. Just as I was getting into my flow. That's all right. We'll get back into it. I moved it to the back pocket. Did I break it? No, I don't. Okay. I was starting to talk to people. Great. Okay, thank you. Can we hear me? Yeah, good. Not? Can you? Yeah, okay, cool. Great. So, Staphylococcus epidermidis. Sorry, okay, I'll lower my... No toxins. It's cool. It's safe. We don't know exactly what they're doing there. We haven't done the functional analyses yet, but this is a really great example of hand taste. So here we see transfer of... It's very, very likely that the Staphylococcus has come from the fermenters' bodies and hands, going into the mesos, living in the mesos, coming to form the mesos. Hand taste. Easy. Here's another example. This is kind of going the other way now. This is not a study I've done, alas. It's an amazing, wonderful study. Some of you might know it. It's by Rob Dunn and colleagues in the US. They gathered 18 professional bakers from 14 different countries in the world, brought them all to Belgium with their sourdough starters, sampled their hands, sampled the starters, they all baked together, they had a great time. They didn't publish that part in this paper. I want them to. Talk about the party they had with all the bread. But what they did find, super interesting, is that the sourdough starters, the microbiome of each sourdough starter, was more similar to, no, let me change that around. There's a better way to say this. The microbiome of each baker's hands was more similar to their own sourdough starter than the hands of each baker were to each other. So what this means is that the most important similarity here is the relationship that each baker is having through the exchange of microbes from the starter to their own bodies, which means that their own microbiomes are more different to those of their fellow humans. You know, going back to David's idea of like blurry lines and this idea of like individuals not existing in nature, like, for me, those are very blurry lines in a great way. This is the other direction of hand taste, right? This is pretty standard. Nothing super new here. The next two examples are a little bit more experimental about how hand taste not only shapes fermentation microbiomes, but also can assemble novel fermentation microbiomes. So this is another story from Kim. I'm mining Kim's brilliance right now in this talk. Thank you, Kim. This is an experiment that Kim ran quite a long time ago, actually. I think like 2016, 17-ish, yeah. Kim was interested in seeing if he could make kombucha from scratch, the dogma of kombucha, both in the scientific literature and in sort of fermentation culture, is that you can only make a kombucha from an existing kombucha, of which, of course, begs the question, whence the kombucha in the mists of time? I can never get over that question. So Kim was like, well, maybe I'll try to make it myself. We know a lot about kombucha microbiomes. We know their main functional groups. Kim also knew that he had a lot of other things in the environment nearby that had the same functional groups. Sunflowers in the restaurant garden, dead honeybees from their hive, his own skin microbiome, of course. He threw these all together in some sweetened tea. And lo and behold, after a couple of weeks, a tiny little biofilm, a little pellicle, a little SCOBY started to grow at the edge of the flower. Amazing. He transferred it to some new sweetened tea. It grew more. After a couple rounds, it was a bubbling, burbling, souring, kombucha-ing thing. It looked like kombucha, it tasted and smelled like kombucha, it functioned like kombucha. When served to guests and presented like kombucha, they were like, this is amazing, it's kombucha, it's delicious. In this example, Kim's own microbiome went into producing this novel thing. Something more deep, maybe, in terms of holobiont hand taste is happening here. And now I come to my last of 4 examples. There could be many, many more, I think, which is a more recent project that we've begun in my group over the last half year or so as part of our larger work into developing more delicious plant cheeses based on traditional cheese-making techniques and using them to study the microbiology of novel fermentations, like how communities from different sources coalesce on novel substrates. And in this project, as part of this project, we're systematically combining different microbes that are typically associated with traditional dairy cheeses, with the novel plant substrates that we're using to make the plant cheeses, as well as microbes typically from the human body, to see how altogether, if we throw them all together in this novel plant cheese substrate, how do new communities form in this novel plant niche? Both with a view to sort of different and potentially better functionality for growth, metabolism, texture and flavor, et cetera, and also hopefully to learn something more fundamental about community coalescence. I'm biased, but I personally think it's super interesting. Super interesting study. This is my postdoc Caroline doing this. So all of these 4 examples show, in their own ways, how hand taste can both help us make more diverse, delicious, sustainable, and place-based foods, while also, hopefully, simultaneously, opening up new, exciting directions for groundbreaking research. And that brings me to holobiont hand taste. So these last examples in particular, I think, show how hand taste is a deeply holobiotic phenomenon. We know that, as we've talked about, our fermentations can shape our body microbiomes, like with the baker's hand experiment. We also know that our hand microbiomes shape our ferments, like the novel misos. This little loopy thing right here, right? We've talked about it. But, however, based on some studies that are emerging in quite different fields of science, generally unconnected to each other, I think that there's a little bit more to this story. So additionally, we know that what we eat can shape our oral and our gut microbiomes. And now we are also beginning to learn how our body microbiome can shape how we actually perceive taste, as well as what we prefer to eat. This is taste up there. Emojis, universal language, right? Everyone gets it. But so far, we don't really know nearly well enough how all of these observations fit together into one big, messy, complex feedback loop linking human and food microbiomes, fermentation practices, taste perception, and taste preferences. In other words, beyond the flow of microbes between our bodies and our foods, this is sort of maybe classic, simple, basic hand taste, we could say, our very sense of what is delicious might also be shaped by these microbes. And this agency, in turn, reinforces their place in our own microbiomes and in the microbiomes of the fermentations we make. And this isn't even accounting for differences in culinary practices and in environmental microbiomes. A lot of this is going to be echoing back to things that David was talking about, for example. This loopiness, I think, for me at least, is a perfect example of a question that cannot be answered by either the natural sciences or the social sciences alone. It totally requires both, and fermentation is a really great way of bringing them together. And just for contrast, we're going to go back to basic hand taste, still cool, very prevalent, but once we holobiontize it, I just made that up now. You know what I mean though, right? It turns into this. Whoa, this is different. And if we think about it this way, I think it starts to open up so many more fascinating, maybe difficult, but also very worthwhile questions to start to ask. I would also say that not only from a research point of view, but if we start to understand hand taste, a phenomenon like hand taste in this holobionty way, it will also have consequences, very real-world consequences, for how we understand and organize a whole bunch of different things like healthcare, diet, agriculture, environmental policy, many more real-world applications, which is also echoing something that Morten brought up in his talk at the very beginning. So I'm going to run through this part very briefly, but I just wanted to really quickly touch on how, as we've heard today, the revelations of the holobiont go very deep. It's reorganizing in a very fundamental way, a lot of basic concepts and assumptions in and beyond the life sciences, with also far-reaching implications for how we understand our place in the world and how we relate to the world, like a lot of what Leonie was talking about. But the concept can also be kind of difficult sometimes. It's like we still have this ingrained intuition that it makes sense to think about things in terms of individuals, well-bounded entities that are citizens and autonomous and stuff like this. So, of course, it's important to do the science and develop the ideas, but when it comes to really deeply, in a kind of physical, emotional way, understanding what it really means for ourselves in our day-to-day lives, that can maybe be hard, making that link. And I think that's where the arts can really help us. So together with some colleagues, also at CEH, and some colleagues at the Medical Museum at KU, as well as an artist duo named Baum and Leahy, we've been exploring that problem together. How can we use a more artistic approach to create experiences and encounters that can help us to, in a more embodied, sensual, or sensuous way, not quite sensual yet, understand what the holobiont really means and is. And this was one such project called Sensing Holobiont, where we tried to combine immersive installation, multi-sensory storytelling, and experimental cooking to explore this idea. And it featured versions of the sourdough experiment that I talked about briefly, as well as Kim's kombucha, the Kimbucha as we call it, as well as a bit of indoor foraging, some soil smells, listening to each other's burbling guts with stethoscopes, a whole host of different flavorful slimes, you name it, it had it. We're hoping to create something, future iterations of this work in the future, so if that sounds at all interesting to you, I can recommend to keep an eye out. We're hoping to do it again. So to finish, I'd like to offer a more general reflection about method. So these days, there's a lot of talk about transdisciplinarity and how we need it to solve some of our greatest challenges. And it can sometimes get lofty, but I also ultimately believe this. I think we really need to sort of try to bring disciplines back together again. It's also really stupidly difficult to do that. And that brings me to one thing that I really love about working with fermentation and hand taste and holobionts. And I think it's one of the things that makes me come back to it all the time in the shower or on my bike or in my idle daydreamy moments. And that's that these things feel like particularly rich sites, at least to me, for trying to bring disciplines and practices and people together, which is something I think we only need more of. And with that, I'd like to say thank you very much. I'd like to thank a lot of people who are very committed, who made all this work possible, all of my collaborators and colleagues who contributed to the work, my team members, Kim and Caroline in particular, and everyone else who's supporting all this work, and of course, Veronica and CH for hosting the event today, and all of you for listening and indulging me. Thank you.